Hello students, welcome to today's session. And in this session I will be dealing with the topic called Microsporogenesis. It is basically includes formation of pollen grains in angiosperms. That is part 1 of the formation of the pollen grains. Part 2 will be microgametogenesis that I will be dealing with the next class. The process of pollen grain formation includes microsporogenesis and microgametogenesis. Microsporogenesis can be described as the developmental process leading to the production of 4 haploid microspores from a diploid microcyte or microspore mother cell by meiosis and cytokinesis. The same thing if we depict in a linear fashion, we can observe that during microsporogenesis, microspore mother cell or MMC, it is also called as pollen mother cell, are formed in the sporogenous tissue of the anther. These cells undergo meiosis to produce tetrads of haploid uninucleate microspores. The process of microsporogenesis may be subdivided into 5 phases leading to the formation of uninucleate haploid microspores. The sporogenous tissue. The sporogenous tissue represents largest cells of the sporangium. The nuclei are hyaline with conspicuous nucleoli and a few portions of condensed chromatin. The cytoplasm represents storage lipids granules which through their oxidation ensure the energy needed to trigger the meiotic process. So the first phase begins with the differentiation of the compact sporogenous tissue. So from the archisporial cells, the sporogenous tissue and tapetal initial will be generated, and the sporogenous tissue now converts or it starts forming a clear-cut compact sporogenous tissue with thin cellulosic walls, and numerous plasmodesmatal connections will be there between these various microspore mother cells. That is the first phase of the microsporogenesis. In the second phase, each microspore mother cell is isolated by a callosic wall in which the plasmodesmata becomes blocked. So the connections between the neighboring microspore mother cells will be lost, and this is the phase where actually the sporogenous tissue starts converting into microspore mother cell. That means from its tissue organization it starts reverse processing to the cellular organization, so it loses all the connections, and in the same phase meiosis 1 and 2 also takes place. In the third phase, it is noted by the formation of tetrads. We can see that tetrads, 4 young haploid microspores that become encapsulated within the callosic wall can be seen, and simultaneously exine synthesis in the pollen wall begins, providing rigidity to the pollen grains. So pollen tetrads will be formed in the third phase of microsporogenesis. In the fourth phase, it is recognized by the dissolution of callose wall and the release of microspores from the tetrad. That means in earlier phase they were together, for spore tetrads were there, but here individual monosporic conditions can be observed because of the breakdown of callose during this phase. And the fifth phase or last phase of the microsporogenesis can be observed by the clear-cut large uninucleate microspore formation, and it is marked by the vacuolation and enlargement of microspore with the completion of exine deposition and pollen wall development. So in this particular session, all these processes will be taking place. So formation of vacuole, enlargement of microspores. So you can see, observe the vacuoles and nucleus, and enlargement of microspore takes place. Completion of exine deposition takes place, and pollen wall also gets formed in this particular condition. Okay, depending on the nature of mitosis, meiosis and cytokinesis, there are basically two types in the microsporogenesis, namely simultaneous type and successive type. And what are the differential features of these two types we will be studying right now. So in simultaneous type, the first meiotic division is followed directly by the second meiotic division, and it is continued by the cytokinesis and callose deposition. Then it forms 4 microspores, and tetrahedral tetrads will be formed. The nature, the type of tetrads formed here are tetrahedral tetrads. It is because the two divisions are formed without cytokinesis. Only after two divisions get nuclear divisions get over, then only cytokinesis forms by the callose deposition, and they form 4 microspores. Whereas in the successive type of the microsporogenesis, dyads with the callose wall forms after the meiosis 1. That means after once the first meiosis takes place, there will be 2 daughter cells and there will be a cell wall formation will be there. And meiosis 2 divisions are independent. That means they are not synchronous. They are asynchronous divisions will takes place. That means since there is a cell wall formation between 2 daughter cells, there will be the further divisions need not be simultaneous. So they will take successive. Then if the spindle formations are parallel to each other, there are so many possibilities of types of tetrads. So we are discussing if they are parallel division takes place between the spindles, then it will be resulted into either isobilateral or linear tetrads. If it is perpendicular, it will result into the formation of decussate or T-shaped tetrads. So now we will study what is simultaneous division. So simultaneous division, there is no wall formation. So the microspore mother cell undergoes meiosis 1 forming 2 daughter nuclei, continued by meiosis 2 forming 4 daughter nuclei. Then cytokinesis takes place. This is the division, the formation of wall formation planes, and as a result there will be tetrahedral tetrads will be formed. Whereas in successive divisions, we can observe after meiosis 1, there is a formation of wall. and in each division is followed by wall. As a result of that, there is a isobilateral tetrad. So if this is the perpendicular division takes place, then isobilateral tetrad can be formed. So if it takes place two perpendicular planes, it will be result into a decussate tetrad, and if it takes place in the two parallel planes, it will result into the linear tetrads, and if takes place one parallel one perpendicular, it may be result into T-shaped tetrads. So ultimately, what happens due to the sequence of occurrence in the meiosis and cytokinesis, each microspore mother cell produces 4 microspores arranged in various patterns, resulting in differently shaped tetrads in different groups of plants. So basically, dicots and monocots, or eudicots and monocots, they will be formed different types of tetrads with respect to, and it is a family character, and to diagnose the family characteristics, this type of tetrad formation will help a lot. Hope this particular lecture was useful to you. Thank you for listening.